Organic molecules is what we're going to talk about next. Organic molecules are molecules that are composed of carbon compounds and have functional groups. We have four major categories: carbohydrates, lipids, proteins, and nucleotides. So, why carbon? Carbon bonds readily with other carbons: hydrogen, oxygen, nitrogen, sulfur. It only needs 4 more valence electrons in order to balance itself out, to form that 8 in its outer ring. And it's easier for it to gain than to lose. So we can form rings of carbon. We can form long carbon chains. And we do these all the time. These are actually, those long carbon chains are a backbone for many organic molecules. The rings are a backbone for many organic molecules. Now, when we're talking about organic molecules, we talk about both monomers and polymers. What are monomers? Monomers are subunits of a larger macromolecule. It is the base form of whatever we're talking about. So if I'm talking about carbohydrates, its monomer is a saccharide unit: glucose, fructose, stuff like that. Or if I'm talking about proteins, a monomer would be an amino acid: tryptophan, for example, alanine, leucine. DNA's monomer would be something like a guanine, a adenine. So they're going to be the smallest we can break down these chains until we start to actually break them apart into further chemical subunits. Polymers are just a series of these monomers bonded together. So a disaccharide, a polysaccharide. A disaccharide is 2 saccharides, 2 sugars bonded together. A polysaccharide is multiple sugar units bonded together. A protein is simply hundreds to thousands of amino acids bonded together. And we call that process of bonding them together polymerization. And this is typically going to be done through a dehydration synthesis process. Translation: we remove a water molecule from off of these 2 compounds, these 2 monomers, and by removing a hydrogen, 2 hydrogens, and an oxygen, we bond them together. And we get a water as well. And that's a dehydration synthesis. We bond them together by removing a water molecule, also known as condensation, by the way. Hydrolysis is when we insert a water molecule into a polymer or another molecule, splitting it apart. So we use a water to split them apart. This is called hydrolysis. All right, so for organic molecules, as I mentioned, let's start with the first one. Carbohydrates. Think sugar, because this is what they are. They're a hydrophilic organic molecule, meaning they love water, and they come in this format: CH2O to the N. So you times each of these elements by whatever N is. So if N is 6, it would be C6H12O6. This is actually the formula for glucose. You will need to know the formula for glucose, because anytime we talk about cellular respiration, aka the creation of energy, we are talking about the metabolism of glucose into energy. As I've mentioned, we tend to call the monomer a saccharide. So if you see a monosaccharide, we are talking about a single sugar. Disaccharide, 2 sugars. Polysaccharide, many sugars. But we also like to use the suffix ose, O-S-E, which means full of. So if you see ose, it's usually an indicator we're talking about a carbohydrate. So here's some of our monosaccharides. These are the simplest carbohydrates. We have three major ones: glucose, galactose, and fructose. And it is the combination of glucose, galactose, and fructose that makes up all of our carbohydrates. One thing to note here, in these rings, in these structures, where you see like here, it doesn't denote if it's an H, a C, or no. Anytime you have a blank corner, like right here or right here, it is implied that that is a carbon. These are carbon rings. Glucose and galactose are hexagon rings. Fructose is a pentagon. Disaccharides are going to be pairs of monosaccharides. And there are three major ones: a sucrose, a lactose, and a maltose. Sucrose is simply a fructose and a glucose attached together. Lactose is a galactose and a glucose. And maltose is a glucose-glucose. Can you have a fructose-fructose? Absolutely. These are the three major ones. This right here, if you're looking at it going, now that sounds familiar, this is your milk sugar. Lactose is your milk sugar. It's glucose and galactose. Polysaccharides. It's all sugar chains. Think chains of glucose, galactose, and fructose. And they come in a variety of different formats. We have three major types of long chains called starches, cellulose, and glycogen. Starch is found in plants. It is produced by plants to help give them support and stability to their structures. We digest it through amylase. Cellulose is another one that is produced in plants to give it support and structure. We call cellulose fiber. And then glycogen is actually an energy storage format found in animals. So glycogen is synthesized by the liver after a meal, and it is a readily available energy source. It's basically glucose chained together to allow for rapid energy release upon demand. Here's the thing. This is temporary storage. This is not a stable storage. This is why most of our energy stores for long-term storage are stored in the form of fatty acid chains, aka adipose tissue. Fat. That's long-term stable. Okay, so what is carbohydrates' functions? The function of carbohydrates is energy. We'll talk about this when we talk about the energy process. Either in this class or any of your other classes, we will talk about the creation of energy from sugar, from carbohydrates. We do also use carbohydrates attached to other compounds to do a couple of different things. We attach a glucose or carbohydrate chain to a lipid. This tends to be found on the external surface of your cell membranes. They tend to help identify the cell as belonging or not belonging. If we attach this glucose to a protein, we are typically, again, going to find this on the external surface of cell membranes, but this is also what your mucus is made up of. So the mucous membranes of your respiratory and digestive tracts, that's glycoproteins. They are carbohydrate protein conglomerates. If we have a proteoglycan, this is going to be primarily carbohydrate dominant, but it will also have some protein, and this is what's going to allow for cells to stick together, cellular adhesion. It's also going to be your jelly filler of tissues, so the jelly-like fluid of your eyes or the lubrication in your joints. That is a carbohydrate protein compound. Moving on to lipids. Lipids are our next organic molecule. These guys are going to be hydrophobic. They do not like water. Think your oil, think your butter, bacon. Less oxidized, and gram for gram, it's going to have more calories or energy than carbohydrates. We have 5 primary types: fatty acids, triglycerides, phospholipids, eicosanoids, and steroids. We'll talk about some of these more than we do others. Fatty acids. What's a fatty acid? It is a chain of 4 up to 24 carbon atoms. However, it will always be in even numbers. You won't have 5, you won't have 7, you'll have 4, 6, or 8. And it is a chain of carbon atoms attached to a carboxyl group at one end and a methyl group at the other. Here's our carboxyl. It's a CHO2, carbon, 2 oxygens, and a hydrogen. And here's our methyl. It's a CH3, 1 carbon, 3 hydrogens. And in between, every group of 2 carbons is referred to as an acetyl group. If all of the bonds are single covalent bonds, it is referred to as a saturated fatty acid. So every carbon has 2 hydrogens. This is a saturated fatty acid. If we have a double bond anywhere, it is now an unsaturated fatty acid. The reason we refer to it as an unsaturated fatty acid means that we call it that because more hydrogen can be bonded. It is not maxed out yet. Now, we like to form what's called triglycerides with fatty acids. Triglycerides are a storage format for energy. Basically what happens is 3 fatty acid tails are attached to a glycerol group. This is done through a dehydration synthesis process, so you will get 3 water molecules out of this, and it will look like this. Triglycerides are actually typically referred to as neutral fats, and their composition will depend upon whether or not we're talking saturated or unsaturated and how long those tails are. The reason they're called neutral is because these carboxylans have bonded, so they are no longer acidic. This will no longer readily give up hydrogen ions. It will not give them up, and it will not take them, provided you don't have any double bonds here. This double bond, it would actually take hydrogen. Hydrolysis of fats is going to occur via our lipase enzymes. Triglycerides are going to be called different things at room temperature. If it's an oil, that means it's going to be a liquid, and this is typically going to be polyunsaturated fats from plants. So double bonds involved. If it's a solid, we call it a fat, and that's typically going to be saturated fats from animals. So liquid oils, polyunsaturated from plants. Think of your canola oil, olive oil, avocado oil. Solid from saturated fats from animals, butter. And again, what is its function? Energy storage. Fatty acids are fantastic for storing energy, but while they're storing energy, they also act as a fantastic insulator and shock absorber for the organs. If you're an individual who is carrying more adipose tissue, you've probably noticed that it takes longer for you to feel colder. If, on the flip side, it doesn't take as much for you to feel warm though. And if you're an individual who doesn't have a lot of adipose tissue, you probably notice it doesn't take much to get you cold. But it does take a little bit more for you to feel warm. It's because adipose tissue is an insulator. And shock absorber, well, you have fat padding around all of your organs that help prevent them from smashing into each other. That's exactly what it sounds like. Phospholipids. Again, we're going to have our hydrophobic tails from our fatty acids, but there will be 2 tails attached to a glycerol. On the other side of the glycerol, instead of having a 3rd tail, we're actually going to have a phosphate group. This is going to give phospholipids what we call an amphiphilic character. The tails are going to be hydrophobic or fat or water eating, and the head is actually going to be hydrophilic or water loving. Let's It looks something like this. This is our classic artistic rendering, a little ball with two tails. And we'll talk about them in a lot more detail when we talk about the cells and the organelles of the cell. But phospholipids are important for many reasons. The primary one being phospholipids make up the cell membrane. They make up the membrane that prevents the cell from being dissolved in its surroundings. It creates a boundary, it creates a barrier. Another form of fatty acid is a steroid. The primary steroid is a cholesterol. In fact, all steroids are derived from cholesterols. Now, I'm saying steroid, and you're probably going, ew, bad. I'm saying cholesterol, and you're probably still going, ew, bad. Here's the thing. Your body creates cholesterol. Your body creates steroids. In their proper amounts and balances, they are not just important, they are vital for your body's proper function. Cholesterol has a huge number of reasons why it's used in the body. It's used in a bunch of different places, but it is going to be responsible for creating a slew of hormones, steroid-based hormones. What are steroid-based hormones? Estrogen, testosterone, progesterone. The hormones that give you your sexual characteristics, that give you your ability to create life. Those are cholesterol/steroid based. Bile acids. What are bile acids? Bile acids are part of your digestive process. They help neutralize the acid coming out of your stomach, and they help break down fats and proteins. So without cholesterol, we wouldn't have steroids, and without steroids, we wouldn't be able to enjoy chicken wing dip, or chicken wings, or pizza, or barbecue. Enjoy your dressingless salad. Yeah. Cholesterol, steroids, super important. Also important for proper nervous system function. So if your cholesterol is too low, we always talk about it being high, but too low is also a thing. If it's too low, you're not going to make hormones like you're supposed to, but also your nervous system is going to start misfiring. Here's the thing though, cholesterol is only produced in animals. 85% is naturally derived from our body. We make it ourselves. Only 15% comes from our diet. This is why reducing cholesterol in diets has limited success with many individuals who have high cholesterol. It's not because they're eating too many eggs or eating too much bacon. It's because their body is making too much cholesterol and not getting rid of it correctly. I'm not saying don't follow the diet. What I'm saying is, if the diet is proving that it's not working, don't beat yourself up. And don't let your doctor beat yourself up. Say you're not following it. If you're following it and it's not working, it means the issue is not your diet. It is 100% your body, not just a 50-50. Now, this is the base of which all of what we just talked about is based on. All steroids are going to have this 4-ringed structure, 3 hexagons and a pentagon. And what's attached and where is what's going to give it its difference. And where these double bonds pop up is also going to be what give them their different names and functions. For instance, this is cholesterol, this is testosterone, this is estrogen. You can see the very subtle differences. The lovely little table putting all of the lipid stuff together. If you're going, whoa, we didn't talk about some of them. That is correct. Eicosanoids we don't really talk about. They're a relatively new discovery. We're still figuring out exactly how we make them, where we make them, exactly what they do. This table here says they're chemical messengers between cells. How are they chemical messengers? We're still figuring all that out. So we're really not going to mention them. Moving on to proteins. Proteins are a polymer of amino acids. Amino acids are their monomer. There are 20 amino acids that we consider essential. meaning we have to have them in order to function. The good news is we can make most of them. Our body creates these amino acids. There are 9 amino acids that we have to have that our body cannot make. We have to get them from our diet. And so this is where you have conversations with people being vegetarian or vegan, or what's the one where they will eat animal byproducts. So they'll eat like eggs and milk and cheese and butter, but they don't eat the meat. So they won't eat the breast meat or the steak or the fish fillet, whatever that one is. We have to have this conversation about whether or not you are getting all of the essential amino acids from your diet. Are you eating a variety of different plant protein sources to cover that gap? Because if you eat a steak, you just covered that gap. You ate all 9. Chicken, pork, whatever the case may be. They have all 9. So that is where this conversation comes in. What makes these amino acids different from each other? One singular thing is different. Attached to the central carbon of the amino acid is what we refer to as an R group. It is the R group that is different. Everything else is the same. The amino group, the carboxyl group, the carbon in the middle, all that is identical on every single amino acid. It's the R group that is different. And the R group is going to be what gives every amino acid its, chemical properties. How it reacts, how it functions is determined by its R group. What's up with these R groups? So these R groups are classified in different categories based on the type of R group. Are they polar? Are they non-polar? Do they have a ring? Do they have a sulfur? Do they have a phosphate? All sorts of different things. So if they're non-polar, they're going to be hydrophobic, water-hating. There's some examples here, tyrosine, methionine. If they have a polar R group, they're going to be hydrophilic or water-loving. And that would be things like cysteine and arginine. But here's the thing. Very rarely do you have a protein chain that is made up of all hydrophobic or all hydrophilic group. Protein chains tend to be amphiphilic. They tend to be a combination of both these philic and phobic groups. All right, some terms here. If we say I have a peptide chain, I am talking about a linkage of 2 or more amino acids. And yes, we do break them down, name them based on how many. If I have a dipeptide, it's 2. Tripeptide, 3. Oligo is less than 15. Polypeptide is the name we give a chain that has more than 15 but less than 100. And a protein is anything with more than 100 amino acids. And how are these amino acids put together? If you guessed dehydration synthesis, so the removal of water, you would be correct. And that is referred to as a peptide bond at that point. All right, proteins have a structure format. They have what we refer to as quaternary structure, meaning a 4-layered structure. They're going to start off with their basic chaining together. That's referred to as primary structure. Then we're going to get hydrogen bonds that are going to form between various groups that are nearby each other, forming our secondary structure. These are things like alpha helixes or coils, beta pleated sheets or folds. Then the R groups are going to start interacting together with each other and with the surrounding water, and this is going to form our tertiary structure. And when we form, or join, I should say, 2 or more different polypeptide chains together, that's when we get quaternary structure. And that looks something like this. A great example of quaternary structure would be a hemoglobin. Hemoglobin is actually 4 chains. 4 chains chained together. And then we have heme groups inserted into them. Heme groups are going to contain the iron and be what the oxygen actually binds to. Just some images. Here's an example of why we care about this. If you've heard of sickle cell anemia is when we have abnormal protein chains. So the hemoglobin is going to collapse, and we're not going to have these good heme groups. What does protein do? It does a lot. Protein is going to give us structure in the form of collagen and keratin. It's going to aid in communication because some of our hormones are protein-based. It's also going to be involved with cell receptors. It's going to be involved with membrane transport, so carrying things across the cell membrane. It's going to be involved in enzymes. Enzymes are catalysts. We'll talk about them later. They're going to be involved with recognizing what belongs and what doesn't, and protecting what belongs in the form of antibodies and clotting proteins. They're going to be involved with movement, muscle contraction. They're going to be involved with binding cells together. They do a lot. Okay, last but not least is the nucleotides. Nucleotides are going to contain 3 basic components. We're going to have a nitrogen base. It will either be a single or double carbon-nitrogen ring. It will have a monosaccharide sugar attached to it, and then 1 or more phosphate groups. For instance, if we're talking about ATP, ATP is a nucleotide, ATP is our energy molecule. We'll talk about ATP a lot more later on, but ATP is composed of a adenine, a ribose, and 3 phosphate groups. It looks like this. ATP has a high energy bond between its second and third phosphate groups, and it is the removal of that third phosphate group that generates a huge burst of energy, which our bodies will utilize to do other processes. Now, that energy production starts with glycolysis. Your body will take 1 glucose and split it into 2 pyruvic acid molecules, generating a net 2 ATP. It can then go one of 2 different ways. If we do not have oxygen present, those pyruvic acids are going to get converted into lactic acid. This lactic acid conversion does not create energy, but it does allow glycolysis to kick over again. If, however, oxygen is present, those pyruvic acids are going to go into a mitochondria. Those pyruvic acids become acetyl-CoA. They will go around the Krebs cycle. Then the products from the Krebs cycle will go into the electron transport chain, ending up with it generating 36 ATP. So all told, a single glucose molecule, if there's oxygen present, can create 38 energy molecules. That's a simplified version of that. We'll talk about in more detail later. All right, some other nucleotides. We have GTP, or guanosine triphosphate, similar to ATP, except instead of an adenine, it has a guanine. We don't really talk about him much, though. Cyclic adenosine monophosphate, or CAMP. It also forms some high-energy phosphate bonds. It typically is a chemical messenger, though, not an energy molecule. And then, last but not least, DNA and RNA. DNA and RNA are actually polymer forms of nucleic acids. DNA is deoxyribonucleic acid. Typically are going to be 100 million to 1 billion nucleotides long and are going to contain our entire genetic code. This entire genetic code is going to be responsible for everything from cell division to sexual reproduction to making various protein chains, so you have hair, eyes, nails, teeth, You digest things, everything. RNA, or ribonucleic acid, is, well, there's three basic forms of it, so they range from 7 to 10,000 nucleotides long. And depending on what form we're talking about, they're either going to carry out instructions given by DNA, or they're going to synthesize the proteins coded for by DNA.